Everybody will fall asleep. Thank you very much, and it's really a great honor for me to give this comments and to celebrate Professor Lloyd's great work. I want to say something. Professor Lloyd, in his talk, said the sentence, "Nature is not a given." And this is what I want to talk about, really. I want to talk, and the subject, as you can see here, of my talk is the biology of culture. So what does it mean? It sounds like contradiction in terms, but of course it isn't. So I want to talk about, very briefly, of course, about 3 points that will make sense of this title, The Biology of Culture. I want to explain how culture changes nature. And I want to talk about how things that we learn and things we do culturally change the activity and morphology of the brain. And give one example. There are many now. I want to say something about, and this is happening in ontogeny during our lifetime. I want to say something about what happens to us within the society and the environment in which we live can change the way our biology works, gene expression, something that is called our epigenetic profile, and I will give a few examples of that. And what is interesting about this thing is that most of the time we're thinking about it in terms also of development, of ontogeny, of what is happening during our lifetime. But we assume this is not inherited. Only genes are inherited, DNA sequences. However, we know today that this is not always the case. And in many cases, in fact, this kind of epigenetic changes, epigenetic variations, which are induced and constructed through environmental and sometimes cultural phenomena, could be inherited. We have evidence for that in animals, not yet in humans, but I'm absolutely sure it will come. And the third thing is something that Danny touched, and that is how, through our cultural activities, we create new selection regimes which lead to a change in genetic constitution through selection. So let me start. I'm starting with a quote by Mithen and Parsons from 2008. By changing the cultural environment, each generation changes the brain of the next. Much of this occurs unintentionally, hence our description of the brain as an artifact of culture. However, when we deliberately seek to influence that developmental trajectory, such as by the provision of particular educational environments or dietary supplements, we are in effect turning the brain into a cultural artifact. What would they mean? Well, what they are meaning is really, they are talking about literacy. And they are talking about the way in which learning to read and write, which has happened happened in parallel in several cultures. Apparently, it was invented more than once. About something like 5,000 years ago. And the idea, and what people are doing now, is they're trying to see what literacy does to the brain. How does it change it? Now, the fact that it happened 5,000 years ago means that it is not the product of genetic selection. It's too short. And for many, and during this 5,000 years, most of the people, by the way, who knew how to read and write were very few. These were the elites. Only in the last 300 years or so is literacy really widespread. So we cannot talk about genetic selection for literacy. And the fact of dyslexia is a very good argument for saying, you know, it's not very well selected, because look at this. We have this variability in the ability to actually read and write. So let us look at this. Now, what we have learned about literacy, I will not go through this slide, I'm just going to, the bits in orange that you see on the brain are the bits that light up in people who are literate when they read and write. When they are shown, for example, letters, which means something, and they read them. When people who are non-literate, but absolutely normal, are shown these letters, these bits of the brain do not light up. So the first thing that we know is brain activity is different between literate people and illiterate people. And not only is it different, it's not spread all over the brain. When you are doing this you are using this technique of fMRI, you see that there is a particular bit of the brain in the left side, on the occipital cortex, which lights up. And it is a very interesting part of the brain with properties that have to do both with the ability to use language and the ability to, for example, recognize faces, recognize visual inputs. Now, the other thing that is very, very interesting, I have a series of quotes here. I want to point to one point which is very, very interesting. And that is that it is not just the activity of the brain that changes. Obviously, the activity of the brain changes when we do things. And when we do specific things like reading and writing, it makes sense that the activity of the brain will change. What is interesting is that it is so specific. Now, the other thing that is interesting is that there are actually morphological changes in the brain. The corpus callosum, the bit that connects the two hemispheres, is thicker in people who read and write than in people who do not. And this is not just something that has to do with maturational changes. How do we know this? Because you could say, well, you know, we teach kids to read and write, and that's related somehow to this. This is not the case, because people were actually doing work in Colombia. Colombia, as you know, there were lots of political problems, and a lot of people went to the jungles and did their bit there, and didn't learn to read and write, and came out when the political situation changed when they were 20 years old, and they were completely illiterate. However, they were cognitively normal people. So they were taken to school. But before they were taken to school, some of them were experimental subjects. They went through fMRI. They were put into the machine in order to see how their brains react to a series of visual signs like letters, and what happens to this brain as they learn to read and write. What happens to this brain in terms of activity and in terms of morphological changes. And what happened to these people who are already 20 years old is that they acquired, through this enculturation process, the right kind of activity, the typical kind of activity of literate people, and also the corpus callosum got thicker. So there is no doubt that there are changes in the brain as a result of enculturation. And we also know the very famous story of the British taxi drivers who have to have a very good spatial memory, because they have to know every bit of London and navigate the streets and so on and so forth. And when their brains were looked at, scanned by the fMRI machines, it was found out that their hippocampus, this is the bit of the brain which is very important for spatial memory, is larger than that of me and most of us here, who are not taxi drivers. So this is another example of the fact that culture changes the brain. It changes it physically. It's a physical, biological change. It has biochemical, and it has very strong biochemical correlates. Right. So now I want to talk about the second part. And believe me, there is a lot of stuff. Since this study on the taxi drivers, people have been put into the fMRI machines with all kinds of abilities, and you know, this is very well established. Okay. Now, here is something nice. I mean, most of the study that we have about epigenetics is the study of changes in gene activity patterns, which are induced usually by changes in environment, in environmental inputs, sometimes also just due to environmental noise. But basically, it is the changes in the activity of genes, in the way that they are expressed, rather in the DNA sequence of the gene. So, and usually when we're looking at what happens to this activity, and we want to look at long-term effects of this activity, usually we stress an animal, really give it a nasty kind of experience, and then see what happens to the gene expression patterns in its brain, and whether this gene expression pattern is afterwards inherited by its offspring and grand offspring. And Unfortunately, often it is. And we know how to measure these epigenetic changes. There are many biochemical, there are different mechanisms of epigenetic, what is called epigenetic inheritance, and there are different types of epigenetic components that can be transmitted from one generation to the next, and we have now the biochemical tools which allow us to actually look at them and measure them. Now here is is an experiment with a more optimistic kind of feeling. That's why I gave it here. And that is that the mice were given, 15-day-old mice were given an enriched environment. And we know that enriched environment is doing a lot of things to the mice, to the brains of these mice. And indeed, and in this case, these mice had a mutation which in normal condition makes their learning defective compared to normal mice. But when they had the enriched environment, in spite of the fact that they had this mutation, it was compensated. They could learn normally. And not only could they learn normally, there were epigenetic changes, but their offspring could learn normally. Unfortunately, it only lasted one generation in this case. But nevertheless, a very hopeful result. So a mutation, a genetic mutation, which usually causes a problem, was compensated through an environmental change. We can't talk about this being a cultural change in the mice. It is a cultural one from our point of view, of course. Now, this is another one which is giving us a more detailed analysis in terms of the epigenetic effects. It's again done on mice. And this one is one of the unhappy ones, the ones where we stress the animals. We stress mothers and offspring, young mice at critical ages, and we are looking to see what happens to them, to this offspring, to this young, as they grow up, and what happened to their offspring and to their grand offspring. So we're looking at first generation, second generation, third generation, and we're trying to see what happens. And what we find is that the stressed mice became mice with lots of psychological problems. Not something very surprising, I guess. If you stress somebody when it's young and you stress it badly enough, this is what happens. So they really get stressed. They really become a little bit neurotic. But not only are they neurotic, their offspring are also neurotic, which is bad news. In other experiments, it was shown that the offspring are not exactly neurotic, but they are more prone to be neurotic. That is, under some conditions of stress, they show they are less robust, less canalized than normal mice whose parents were not stressed. And we know, in this case, much more about the biochemistry. That's why I gave it, because it's a beautiful example of a very important work where we are beginning to uncover the biochemical details of what is going on. And we're learning more all the time. Now, the last example that I want to talk about, and what I want to say, before I go to the last example, what I want to say about this is, one, we have examples in humans where we know that there are epigenetic changes in the brains of people who are extremely stressed as young children. For example, people who committed suicide because they were sexually abused as young children. And when compared to people who committed suicide for another reason, there are many reasons to commit suicide, unfortunately, but when these two groups were compared, those who had this very severe sexual abuse had different epigenetic profiles in their brains. And this was 20 years later. We also know that the effects of things that happen in in humans, that the effects of what happens during pregnancy, the pregnancy of the mother, for example, starvation, has effects on the children, on their children, and on their, to some extent, lesser extent, there are beginning to be studies that show that there are also some effects on their grandchildren. It's going down. But it's not just one generation effect. So again, and here we're talking, for example, about wars, very, very culturally induced kind of phenomenon, the stress of war. We're talking about the case of the starvation of the Dutch population during the Second World War, when the Nazis stopped food arriving to Amsterdam and the area, and the people were living on something like 600 calories a day, and pregnant women were also starving. And since in the Netherlands we have very, very good health system and everything is registered, and so one can follow this, and we know that the offspring of these mothers in different trimesters of their pregnancy had a higher risk to develop all kinds of diseases, from heart diseases to schizophrenia. And I think that it is very important. Unfortunately, we live in a world in which there are a lot of problems of starvation, of war, of chronic stress, and to see what happens, not only to the generation that suffers through it, but also to the next generation, is very important. And maybe, who knows, it will convince politicians, I don't really believe it, of course, to do something about it. Right, so now I want to talk about the third example, the third aspect of the way in which culture changes biology. And this has a lot to do with what Danny was talking about, but I will give a simpler example, and a well-known example, the example of the spread of something that is called the lactase gene. The lactase gene is a gene that allows us to break lactose, the milk sugar, into its ingredients and use milk as an energy source. Now, this spread of this gene is a recent phenomenon in humans, and it is related, we know, now, not every population, by the way, has it. It is common and very frequent, the frequency of these genes, in those populations that use fresh milk. And it is associated strongly with the domestication of cattle. So what we know here is that dairying was, the domestication of cattle occurred something like 7,000 years ago, something like that. We don't know exactly, 7,000 to 5,000. Dairying was practiced in some populations, not in all populations, and dairying and the use of fresh milk was practiced in a fraction of the population that actually used milk. In some populations, milk was only used as yogurt or as already processed product, not as fresh milk. Now, what we see is that in those populations that used fresh milk, there is a high frequency of the gene for breaking lactose, and and there is a lot of work about it, but the work sort of converges on the fact that it is correlated very strongly with the domestication of cattle, and it is the domestication of cattle that preceded the selection for the lactose gene. So the domestication of cattle created conditions where people in some populations and for local reasons used fresh milk. Because they used the fresh milk, there was a selective advantage for having the gene who can break the lactose and can use this as, and the milk can be used as an energy source, because normally, in normal people, once we are weaned, once we stop drinking our mother's milk, this gene is not expressed. Right? So the gene is not expressed normally, except in those populations, now in many populations of the world, but not all, where it is, when the gene is expressed, and this is the result of mutation. So this mutation spread as a result of culture. Culture created the condition for the spread of this mutation. Now, a very similar thing is happening with respect to something very interesting, sign language. Sign language, which was introduced to the United States about 200 years ago, has changed the genetic constitution, apparently, of part of the population. How did it do it? Right. Before sign language was introduced, people who were deaf and mute were considered to be retarded and were treated as retarded. They didn't, very often, they didn't marry, they didn't produce kids, and their their fitness, if we talk in biological terms, was rather low. Now, as a result of the introduction of sign language, two things happened. First of all, these people were treated more as human beings who can be mates and who can be partners in all domains of life. And the second thing was that people who were mute and deaf tended to marry other people who were mute and deaf because with them they could communicate best. Now, the most common type of mutation which leads to deafness, there are at least 30 of them, is something called connexin. That's why it's called connexin deafness. And as a result of deaf-mute people marrying each other, the frequency of connexin deafness went up. Why? Because the connexin deafness is what biologists call a recessive mutation. You need 2 copies of it in order to be deaf-mute. If 2 deaf-mute people who are likely to have connexin deafness are married, the chances are that their children too will be married. And therefore, connexin deafness is going up in the population for 2 reasons. One is this kind of non-random mating between connexin deafness carrying gene, people who carry gene, and because deaf people are treated like human beings and as potential mates. So this is a very, very cultural change which changed the genetic constitution of the population within a very, very short time. Here is how quickly, when you're looking at the graph up there, it's a theoretical graph based on the data. You can see how quickly it has changed. And this is something to do with the evolution of language that Danny was alluding to, and that is that, again, what is happening when we're thinking about a lot of the kind of things that happen, the thing that we call cultural, and in general the thing that we call cognitive, is that the plasticity of the brain and behavioral plasticity is enormous. It goes beyond what was selected. Here we have Kanzi, a bonobo chimpanzee, who has been taught to communicate through symbolic symbols, through lexigrams. Now, I'm not going to go into the discussion of whether or not Kanzi really knows language or doesn't know language. It's not of interest to me at the moment. But what I want to say is that for sure Kanzi can do something that its mates in Zaire, who have not been taught to read and write, to communicate in this way, cannot do. We can stretch the plasticity of the brain, and then the genes can follow. Culture and changes, plastic changes in development, usually, not always, but usually precede genetic changes. Genetic changes are the cart that runs behind the genetic, the developmental horse and the cultural horse. Thank you.